This is a production of Cornell University.
Well, welcome. I'm so glad. This is a lot more people than I expected. I think the last time I did a talk here, it was like 2 people. It's like during the peak Zoom time. But this talk does work so much better if we're standing in the orchard and I can like do the Willy Wonka, like look at paradise, like look at this beautiful diversity collection. So you're so close. You're like so close to paradise. Geneva's not far. Like we do tours, public tours. Marvin, like we do lots of tours for horticultural groups from Cornell. Like take a chance to come up and see and taste fruit. And that's really how like germplasm is meant to be interacted with and discovered. So I am the curator for the apple and tart cherry collection. I'm going to talk about the grape collection that's up there a little bit, but I'm really grateful for this opportunity. Let's see, here we go. And of course, like I'm the only one talking today, but this is all like a team effort, really. So we have, a core of 3 scientists on the team, and also Ganwan Zhang, who's the research leader up there. But like we have tremendous expertise in grafting, in chemistry, in pathology, entomology now with the new addition, Michaela Robertson. And who can forget interns, right? Like this is kind of the highlight of the job. And I really like advocate strongly for anyone who's in the science, like mentor as many as you can, like find your replacement, right? Like we can't do this job forever. Like you need to get the next generation motivated and trained to kind of step in and take the reins. So we've done, maybe 23 interns now at my time starting in 2018. And again, this is, I think I always give this quote from Aldo Leopold is like kind of my, like the philosophy that I try to like frame all of our discussions in horticulture, agriculture. And when we're talking to broad audiences, maybe this isn't the audience for this quote, but I think this is useful in connecting to the next generation, to connecting people who don't think that they're part of agriculture. But really there are 2 spiritual dangers in not owning a farm. One is the danger of supposing that breakfast comes from the grocery, and the other that heat comes from the furnace. And I do think that this part of New York does a little bit better job at that than most of the country. But still, there's a lot of work to do. And really, like when you consider about 2% of the US population is directly involved in agriculture or ranching, like there's a lot riding on the few that are contributing to this. And this is how I feel. This is how maybe it feels to all of you working in your respective fields. Like we're Sisyphus pushing this boulder up the hill, and like, other fields I don't feel like have this challenge that we do, right? Like if they never made another smartphone, I think we'd adapt, right? We'd be fine. It's not like once they stop making smartphones, we roll back to like the rotary phone, right? But if we stop the research and the progress in agriculture, there really is a threat that we roll back production. We roll back that sustainability as new pests, new challenges emerge. And like, really, there's no end to what we need to do, right? So really like passing that boulder off to the next generation so that food security is like, not the pressing concern for our society, right? Like we can solve other societal issues rather than just trying to feed ourselves. But not all doom and gloom, right? Like this is the flip side. Getting to work in horticulture is that there's a joy and there's like a childlike, wonderment at the diversity that's out there and the exciting discovery. So hopefully, so this is not the original Willy Wonka. This is Tori Meekum, who's our molecular biologist and a talented intern of ours, did some nice Photoshop. But actually, I do need to update this because I mentioned that to an NPR in an NPR interview with Susan Brown that Susan's kind of the Willy Wonka for apples, and they picked up on that and published it. Now Susan Brown kind of gets teased a little bit for her Willy Wonka-ness, but there's a joy and like there's mystery and some, I don't know, impishness to like coming to visit the gene banks and tasting weird flavors. And not everything will pass you is desirable. And hopefully this group is well versed in germplasm. And I don't know that there's like maybe a course or thing that like really hammers this home, but like what is germplasm? And it's just simply like bearers of heredity, right? Like these living resources that can contribute to plant improvement. And it can be any number of things: modern cultivars, heirlooms, land races, crop wild relatives. Like we do a lot of work with those. And I think I highlight here the value of these crop wild relatives, because all like of the really exciting traits that like, I lose sleep at night thinking about like how cool some of these traits are. The disease resistance is huge for sustainability, but flavor, like Concord, the kind of the classic grape flavor comes from a wild grape. And what we associate, with the candy flavor of grape really is like a wild trait that is introgressed. Strawberries, right? Like the production of strawberries, like that ever-bearing trait comes from a wild strawberry species from Utah, where I'm a native too. So really like getting excited about, bringing in the crop wild relatives, like the land races have a tremendous history and story. Like this is the story of civilization told through agriculture. And it's so important that like, not just speaking of them as tools that we can use to make new cultivars, but really like preserving our history and our culture. And really it's this balance, right? Like, so the breeding pipeline is to like come up with amazing cultivar, but there's the selection process really does narrow down that. And like, really this is showing the breeding. I don't know if you can see my pointer. Is that coming through? Yeah. So like the breeding is leading upwards, but really like that's just poor design. Like, cause the breeding cycle is going back through each layer and trying to bring in new traits and new to maintain this broad diversity. Because again, no breeder worth his salt or his or her salt is gonna like breed themselves into a corner, especially for apple breeding, which again, the generation time, like to be able to introgress new traits and new germplasm is gonna set you back quite a bit of time. So always be thinking like, how can I maintain this while trying to achieve, broader and more, broad diversity and targeted releases for new cultivars. So yeah, avoid the bottlenecks. Because that's really kind of evident here when we do genetic studies of our apple collection. Each line represents a first degree relationship. And so in our diversity collection of about 2,000 apple accessions, you can see how everything's kind of already intermixed and somehow related to one another. But apple, because of its biology, really retains a lot of diversity, but modern cultivars really only have about 8 like progenitors. That is like the effective population for apple. That's kind of scary. So I am part of the National Plant Germplasm System, which is a USDA program that has about 20 sites throughout the US dedicated to crop conservation. Geneva is unique because we are one of the original 4 that started in the 1940s. So here's Geneva here. I'll highlight. Again, here's like the overall holdings of the entire program, about 621,000 accessions. And again, this isn't just like a cool collection, like an ARC that no one has access to. This is like broadly accessible for research and for breeding and for commercial development. So about 20, a 250,000 samples go out every year from our gene banks, and that's pretty amazing. And with our simple mission of acquire, preserve, characterize, and distribute these genetic resources. I do love this quote by the geneticist Francois Jacob. Diversity is a way of coping with the possible. It acts as a kind of insurance for the future. And when people get excited about talking sustainability and how do we like preserve, agriculture for the future, that genetic diversity is a part of that solution. So our gene bank does look a lot different than maybe what you've seen. Like, I know Denise like has a lot of experience working in the classic gene bank, but here is our gene bank, and it's really like not preserved at all, like not protected at all. Like, so we have deer issues, we have disease management problems, just because these are field-based collections. And you can see we do have, a substantial diversity of apple and 56 species with about 4,400 accessions in there, making it one of the world's largest and most diverse genetically in the world, which is really exciting. The grape collection has about 30 species with 1,400 accessions, and the tart cherry, which again is it's small, but it's one of my favorite things ever to talk about. So here's just kind of our numbers. We mentioned like the 250,000 distributed. Geneva mostly was distributing apple for a long time, like reaching about 6,000 to 8,000 accessions a year, or samples a year that we were distributing. Post-COVID, we've had some pullback just because we wanted to focus our energy into making sure that germplasm is targeted for breeding and for legitimate research. So here's kind of the classic view of like, this is a gene bank. Anyone recognize the one on the left? Where is it? Svalbard, yeah. So like the Doomsday Vault, right? And so we have a vault like this for our vegetable and seed collection. Hemp, I should mention as well, is in a vault like this. And this is what I mean by we need to balance vulnerability and accessibility. So apples are easily, like the simplest way that people have propagated and traded apples over time and preserved them is through clonal propagation. So you can keep seeds all you want, but if you want a clonally maintained line, like a pure Honeycrisp, it's only through clonal propagation that you can do so. And so if you want it accessible for pollen and you want to do crosses, you need a mature tree living in the field. And so in 2020, we had an outbreak of fire blight that really just devastated each square here in the grid, our field map represents an accession, and the color indicates disease severity, and purple being the most severe. And it's kind of, there's a lot of purple in there. So at a time that like we were reduced workforce because of COVID policies, we had this outbreak of fire blight, which really was a challenge for us. But hey, we got on the cover of Agronomy with one of our pictures. So we did an evaluation, just let's go through the collection and just evaluate, like let's do a quantitative score of how infected these trees are just by counting number of fresh and new growth, and then taking account of how many infected shoots and just giving kind of the rough percentage of those. And you see quite a bit of broad variation, from 0%, which could be just a lucky escape, or I like this one, it's like 64% here and about 0.9%. So they're right there, we're, a high probability of tree, C here getting some disease pressure, but not showing any signs of resistance. And then when we map like the results by species, you see like a lot of the domesticated apples have, kind of interesting broad range of no infection to heavy infection. Mouse severcii is the wild progenitor, so it kind of makes sense for that one as well to have a similar pattern. That was June. July, you see a lot of those healthy trees disappear as disease progresses and we start to capture more infection. But a lot of the wild species retain their resistance to the disease. And again, the challenge for us is maintaining these collections is there are amazing horticultural practices, a lot of them developed at Cornell to manage apples, but not diverse collections of apples where everything might bloom at the same time or at a different time, which is critical for managing a disease like fire blight or the response to a certain pesticide. And you might be, sometimes we were doing more damage than good by putting on certain applications of copper, right, for example, or prohexadione, apogee not each accession doesn't respond to the effects of apogee to reduce growth, to kind of limit the disease. So it really is a tightrope walk of orchard management to like hold all these diversity collections, keep them all together while managing all these different, challenges and requirements. And so a lot of pruning and regeneration happened during that time. And I think our total was close to 3,000 individual trees were propagated and replanted in Geneva. And we're doing so much better now with a diverse management program, chemically, that is. But there's still an impact on fruit quality. Again, this is where we don't rely on commercial practices. We can do things that like will sacrifice fruit quality to maintain tree health. But this is one of the consequences is like we'll see heavy russeting, heavy splitting, which makes the fruit kind of not useful for characterization, which is a critical part of our mission. So we have these healthy trees, right? Or we have a wonderful diversity collection that no one's interested in using because they don't know what we have, right? But during this kind of heavy fire blight time, we couldn't do a lot of characterization to take representative images or get good fruit quality data from those. The other kind of safety net that we utilize heavily is cryopreservation, and apple is one of the easiest to cryopreserve. It's simple, collecting dormant sign wood like you would right now for grafting, and you can dehydrate it and put it in liquid nitrogen. And almost the recovery is, 80 to 90% viability, and it's tremendous. It's really easy to manage and get your diversity collections backed up this way. Grape is a lot more challenging. It doesn't direct cryo. There's like a shoot tip and a droplet vitrification. So going into tissue culture, getting droplets, freezing those, and then bringing those out, right? So you can imagine that the labor intensive part is like kind of the bottleneck there. Again, clonal propagation, this is one of the coolest and most ancient tools that we have, right? Like dating back maybe 10,000 years of people demonstrating that this capacity. So this is likely how apples were taken out of original forests and traded and brought to your backyard, right? Like no more foraging. Now we have cultivars we like, we can put them right where we want. And so again, just in kind of an example of how we do grafting and propagation to maintain our collections. And so these are examples from cryopreservation, which you can see here's the rootstock, and then here's the chip budded accession. And then yeah, you'll get some growth, and then you can trim back the rootstock and have a brand new accession. And what's so cool about this is now you have a timelessness to the diversity. And so things that are hundreds of years old or sometimes thousands of years old, they report some grapes dating back to Roman periods. Roxbury russet is a cool apple in our accession because it represents the first true American apple. It's this kind of wild triploid thing that's not that tasty, but it's part of American history. Montmorency dates back to the pre-17th century and represents the largest cultivar grown for tart cherry production in the US. And our buddy, Concord, right? Like we love this grape and its distinctiveness, but this is an old one as well. And so what's amazing is that you walk into these collections and you can see things that were developed, within the last 10 years or like the last several hundred years in our collection. And they're intermixed with the wild species as well. So you kind of get this snapshot of all the diversity in one time. So now I'll just walk through some of the exciting, I think they're exciting characterization efforts that we're going through right now. Because again, it's not enough to just have these things be secure. We want people to use them. And one of the limitations to people having access is just they don't know what we have or what would be interesting to them. So collecting phenotypic, genetic data, like getting that information out, like maybe it's got an interesting pedigree or a history, and that makes it more accessible. So the tart cherry is kind of a nightmare though genetically. So it's a tetraploid hybrid of a sweet cherry and a wild European cherry. But it's got this really interesting anthocyanin profile that it's like, so here's the anthocyanin. Again, it's not just red, right? Like it's a different profile, which gives it different chemical properties and distinguishable through chromatography. Kind of interesting that you can tell a sweet from a tart just by its chemical fingerprint. Melatonin is reported high, and this is one of my white whales for characterization because it's just such a fascinating compound to study. Thank heavens that TikTok was saved, right? Like, I don't know if you guys were nervous about that, but how else would we have known about the Sleepy Girl mocktail from tart cherry juice, right? So here's a great use of social media to promote a crop. Go and look this up. Again, there's a lot of interesting, anecdotal things that like we do with our crops. But Sleepy Girl mocktail highlights kind of the sleepy benefits of tart cherry juice, which might be related to melatonin or might be nothing at all. But we'll investigate more. What's cool about tart cherries is that they're kind of having a moment right now. So I guess I would say like grandma drinks prune juice, right? And like your ultra marathon runner is chugging down tart cherry juice. And so like they have like, this is one brand, like they've got the pit crew. Who doesn't want to be part of the pit crew, right? That's great. That's great branding. And it's like a great promotion of a product and it gets it into diverse markets and applications. And it's not just a $3 bottle of cherry juice now. It's a $15 bottle of elite, endurance juice, right? Like drink this if you want to gear up for, I don't know, Marvin's seminar or like maybe he's got a tough exam coming up. Chug some tart cherry juice, maybe it'll help. But the problem with the tart cherry industry in the US is it's like precariously dependent on a single cultivar. Which again, if you take any agriculture class, you know that this is bad news and you shouldn't do that. So we wrote a fruit quarterly on like, let's move beyond Montmorency, highlighting some of the interesting diversity within the USDA collection. And I highlight this again because you can see the red is our tart cherry and it gets very little attention compared to, again, it's maybe $100 million industry in the US on a good year compared to like the billions that grape and apple bring in. So it's nothing. It's not the smallest crop, but it could benefit from some diversity. So we've looked at phenological traits. So like bloom time alone sets back the industry every couple of years just by a frostbite or frost damage and ruining your crop. Fruit chemistry, I mentioned like interesting phenolic profiles that like might be related to some of the nutritional benefits of the juice, and then fruit, just generic fruit descriptors and images, pit weights, looking at pit to fruit ratios, so you can like kind of maximize fruit size and not waste energy and a market on a heavy pit, this large, excessively large pits. Looking at tart cherry bloom phenology, we do see some significant variation. So this maybe represents a month-long span, with the vertical line here representing Montmorency. So you can see there's some extreme late blooming beyond Montmorency, and then actually we could do a lot worse for New York State. And so where the most of the production is in Michigan State, which they have maybe an even more volatile springtime than we do some years. So again, here's an article from 2012 when I started at Cornell, and I wondered we talk about tart cherries, where are all the tart cherries? Well, there was a frost that year that killed Michigan and Geneva's tart cherries. So beyond just like looking at bloom phenology, we can assay using differential thermal analysis that Jason Londo, now at Cornell, is a specialist in. And so we did a 3-year evaluation of 64 tart cherry accessions, looking at its cold hardy adaptation, acclimation and deacclimation over time. And we're excited to like finally summarize these results and get them out. But just some initial graphs, you can see that each species, and then within the species, there's variation for this trait, which again is going to hopefully help us target climate-specific cultivars, or if there's a new region that wants to expand their cherry production, how it could be done. I do like this image of tart cherries because other crops have these beautiful diversity displays. Like tart cherries kind of all really do look pretty similar. Like there's not a whole lot of like morphological or size variation that exists currently. So it's maybe a gap for us to fill. But each one of these like box plots represents a different trait. You can see total soluble solids, the sweetness, right, or its acidity, or its acid sugar ratio. And each one, like Montmorency, is kind of on the low end. So low end for sweetness, but not bad for its sugar acid ratio, which is actually more important than the total content to begin with. It's just our perception of how sweet or how acidic something is. I think this is what sets back our whole industry, is the weight of Montmorency is pretty good. And it's like, it's on the late blooming, so conversion of an orchard or like adoption of a new cultivar is gonna be kind of hinging, like, can we get the same fruit size, the same dependence, like, I don't know, the same quality from a new cultivar. Even though if you look at the anthocyanin profiles, which again is the new market for tart cherry juice, is like high nutrient density, like it tends to be on the low end for almost every trait. So here's an example. We're kind of chromatography nerds in our group. So again, sorry, apologize for the excessive use of chromatograms, but here's, again, the specific tart cherry anthocyanin and then the sweet cherry anthocyanin. And again, here's a sweet cherry. It's missing mostly this glucosyl retinoside compound. And then you do see this kind of interesting hybrid-like ratio split between the two. But again, Montmorency kind of on the low end for the content variability. Aroma, and so I get really excited about flavor and aroma diversity within the collection. So benzaldehyde is the classic cherry compound that we associate with candy. It's also can fool us to thinking something's almond as well. It's the same chemical. But here's like some benzaldehyde results, and you can see Montmorency kind of on the low end. So you could really drive that cherry flavor by using different diversity or for breeding purposes. This one was an interesting hybrid accession with another wild species, and it has this more common grape aromatic, linalool, which kind of is more floral in flavor and almost missing benzaldehyde. So you can kind of see as you start to profile like where you could push flavor in a breeding program, or even just using these as direct to market for an heirloom production or for something else. But so linalool content really is exciting to me. So we have seed friends in Geneva that do a lot of seed analysis. So we can use their equipment. So here's a Marvin seed analyzer. You can throw pits on and a whole fruit because cherries are small enough, and get circularity. So we're not spending a tremendous amount of time kind of with calipers and doing these individual measurements. So it's kind of a nice higher throughput Genetic markers are invaluable, but challenging to like use for breeding just because of the tetraploid nature. But you can see a clear split between the sweet cherries in red, the tart cherries in black, and the progenitor fruticosa in green here, and some kind of like interesting things that, yeah, are happening in there. I won't go too much into that. Kind of the highlight, though, of my career, I think so far, has been this discovery, Mount Vernon, the home of George Washington, right? For the history buffs in the room, right? Like this is like one of our founding fathers, the first president of the United States. They found cherry preserves during an excavation of the basement there. And they found, initially one bottle. We got a phone call because like they knew it was cherries from the pit specimens. And how do you ignore that phone call, right? Like we went down immediately while they were discovering bottles in the ground. This is Tori and I, like there at the dig site, and they were like, everyone was screaming out, "Oh, we found a bottle 27." And it was like kind of a thrilling Indiana Jones moment, right? And are we going to unleash some curse, or it's like, I don't know, some interesting horticultural artifact? But so Tori and I actually have the privilege and honor now to be sifting through the contents of those bottles, pulling out pit samples, which we can compare to modern pits and look at, like, because again, the pit seam and the pit shape all kind of is indicative of a cultivar. Here's some stem fragments, which like that's amazing preservation for about 250 years old. Here's like kind of like a bottom like or a top down view of one of the bottles, which again is like some of the fruit is still intact and exciting. We've isolated DNA from them to compare to a reference library, and it's, yeah, super exciting to be able to kind of bring to light again one of like, this is a huge American story that we get to be part of the telling. So here is moving on to grape, and grape is interesting because it's split between two sites. So we have a tale of two collections, one in Davis, California, one in Geneva. I don't know where you'd rather be today, but I don't know. California seems pretty nice, minus the horrible fires. But so primarily vinifera, or the historic European wine grapes, table grapes in California, and we specialize in cold hardy hybrids, which again, it's like, we call it the cold hardy grape collection, but it's so much more than that. Like disease resistance and flavor is what we really should be promoting. So we're doing a large profiling of about 350 vinifera accessions and about 100 plus hybrid accessions from Geneva to look at like anthocyanin profiles, which are huge for, again, nutritional content, but now wine and table grape industries. These are just a few of the like interesting profiles that come out. Like, so these are all run with the same analysis. You can see some anthocyanin rich, some really anthocyanin poor accessions in melon versus royalty, and the classic Merlot, right? Kind of like in middle ground there. This is Papa Smurf. So this is, yeah, it's our workhorse LCMS in the lab there. So again, when we look at total caphtoric acid, which is important acid in wine, we see again some extreme, with Merlot being on the really low end, and some really interesting cultivars or accessions on the high end, which some studies are showing caphtoric acid associated with other physiological traits like maybe drought tolerance and some other things, which will be kind of interesting to compare. This is with fruit, but what do leaves look like as well? Again, in anthocyanins, the color of your wine is so important, not just for like anthocyanins, but for the marketing. Merlot's kind of, again, on the middle to low end, not of the box plot, but we do have some really extreme high anthocyanin cultivars. And again, these are just some interesting stats from our diversity evaluation. So one cultivar, royalty, had 30 individual anthocyanins. Anyways, we talk about like, how do we diversify the human diet, right? Like, so anthocyanin rich foods, but anthocyanin diverse foods as well play a major role in that. We do solid phase micro extraction, or SPME, which we're using a method developed by Gavin Sachs, the famous wine food scientist here at Cornell Ithaca. And it's super low input platform that gives us a lot of really powerful results. So again, here's our Concord, our Foxy aroma. So we were able to, all these purple compounds are ones that are heavily associated with the Foxy aroma in grape. And so we can profile that. And Muscat, I mentioned the linalool, so Muscat grapes have just such a distinctive and lovely aroma. like a French perfume. I don't know how else to describe them, but better, but sweeter. You don't want to drink French perfume maybe, but these grapes really like have such a fascinating flavor, mostly from linalool and other monoterpenes. So again, that's why I'm really excited about our tart cherry that has more of a muscat grape profile. And here, if we're looking at linalool diversity, this one accession is really on the high end. July muscat for this nerol compound, another similar or a muscat associated compound. One of the challenges is like the name. So we have the cold hardy grape collection, which is more than cold hardy, full of hybrid grapes, which could be anything. It could be domestic to a wild or wild to wild, interspecific, but it's just this nondescript term that really makes it hard to leverage the power of the collection. So here we have stout seedless, which is 90% vinifera, and Napka, which is 10% vinifera. And Grin in our database are both called hybrid. So pulling out that information really limits, right? Like if you're searching just by taxonomy alone. So we did ancestry estimations using PCA, principal component analysis, and you can give a percentage to how much of one particular ancestor contributed to one or another. So we have now kind of a continuous variation of from 0% hybrid to, or 0% vinifera to 100% vinifera in the collection. So at least that like classification helps bring to light some of the cultivars that we can use. And these are just, I always have to highlight this because Peter Cousins, who was a longtime breeder for the USDA, now at Gallo, has developed these cultivars from hybrid grapes in Geneva. So like high disease resistance, really interesting environmental adaptation, but also amazing fruit quality as well. So a kind of proof that you can take something hybrid with a wild background and bring it into a more cultivated and not just serving New York industry, but California industries as well. Kind of the heart of the collection. This is what I spent my PhD studying, is the apple collection and working with Susan Brown, the apple breeder here at Cornell. But we've got, several thousand accessions. And when we talk about an apple a day, this is almost, 15 years of apples a day if you were to try one accession every day. So kind of endless joy, right? If you're looking for that Wonka everlasting gobstopper, like you can just spend years searching the collection looking for that. This is kind of showing our footprint of distribution. Like we've really touched a lot of the major apple production world in the world, which is kind of cool. Like, so these are accessions that landed somewhere internationally or not. And apple has an interesting domestication because we talk through grafting, this instant domestication, but apple itself is a hybrid between two species. And this story has been retold and told so many times. Every time I want to gloss over it though, someone, maybe Marvin in a tour, like, oh no, you got to tell the story, and have to go back and highlight that. But so really trading apples out of Kazakhstan through Europe where it hybridized with a different apple. So we get this interspecific nature to apple, which makes it interesting. Any guesses on which is the domesticated, which is the wild row? Top, middle, bottom? What do you guys think? Middle. No, middle? Yeah, you're actually right. So that's the Malus severcii representatives that we have in the collection. Way to go, Hannah. And so this story really was famous. Here's Gennaro Fazio. I love this picture of him. And the first curator for Apple, Phil Forsland. So they got the helicopter in to Kazakhstan. How cool is this, right? Like I'm talking Indiana Jones level, like just going into a basement and looking at old bottles and stuff. Like they actually got a helicopter in and collect fruit from the wild, which is really cool and was popularized by Michael Pollan's The Botany of Desire. So this is showing over time the growth of the apple collection. You can see starting with a large donation from Cornell, the breeding program, a lot of wild material, and then huge amounts of Malus severcii, the wild progenitor of apple, coming in the 90s. And over time, it kind of has like slowed down to like being more targeted for wild species. But the story really wasn't done being told until we had strong genetic background and genetic data, because what we thought was pure Malus severcii, so we brought back maybe 383 accessions into the collection. 151 only were pure, what we considered pure species, pure Malus severcii. 215 were admixed, so we have a hybrid population, and then 17 were pure domestica in the forests of Kazakhstan. And again, explain how that can happen is kind of amazing, right? So apples get around and they're well designed even through propagation to propagate themselves and just be planted where maybe they shouldn't be planted. But this is kind of highlighting the really, I don't know, the stress and strain of like collecting wild apple today and not waiting, right? Because if there's an apple orchard down the road, that pollen is going to admix into a wild population. And over the course of, a few decades, who knows what will be left of that. So the genetics are secure with apple. The purity of a species, if that's a primary concern, is kind of a nightmare. What's so cool about our story with Malus seversii, though, is all of this came out that it was still a mystery of who was the wild progenitor of apple. And it was germplasm found by the USDA and brought here that has been now distributed to Europe, back to Kazakhstan for repatriation. And it was like really that initial effort to conserve Malus seversii that gave us so much scientific information. So it's kind of, it's a huge win for our unit and the legacy of Phil Forsland and those Herb Aldwinkle. You have to mention him as well. So when we look at genetic diversity, though, within the collection, we see a lot of clones because a lot of things have names and the names change. And so we bring it in not knowing that it's actually identical. Sometimes the clones are like take up space, but sometimes they're really useful because you might have just single mutation sports that like maybe it's Golden Delicious with a new novel trait added. And that kind of is exciting for breeding because otherwise these cultivars are stuck without any kind of mutation just because they're fixed in time through clonal propagation. Of course, I have to show more chromatograms. You thought I was done. But this is licorice flavor, estragol. And so apples, most people don't have this familiarity with apples, but if you go to an orchard late in the season and harvest even a Golden Delicious, you'll get a bright licorice flavor. Maybe it's dark, depending on if you hate dark or if you hate licorice. But it's a fascinating flavor, and I think it adds to the diversity and what consumers want in that novelty. I think the red-fleshed apples maybe had their moment, and it's not that exciting anymore because it didn't really bring a lot of interesting flavor or interesting, I don't know. We love that element of surprise, and just red flesh wears off pretty easily. But flavor, I think, is kind of a powerful target for modern apple breeding. Alpha-farnesene is kind of associated with some disorders in post-harvest, and you can talk to Chris Watkins about all of those. But Honeycrisp is one of the major, I don't know, the troublemakers with some post-harvest disorders like scald, and alpha-farnesene is associated, but I don't know if it's really clearly demonstrated how it is, but this particular aroma is really high. It's got some estragol, but low in this particular sample. I have to leave with some life advice. So wherever you go, maybe you don't care. Maybe you've tuned out apple, grape, and tart cherry this whole time. Maybe your thing is carrots. I don't know, or berries. But like just whatever trait, whatever crop you're working on, just consider the germplasm. Like so many studies that I read from a curatorial or gene bank perspective, like fall short of what they could be, had the authors more carefully considered what diversity that they were including in a study. And again, you can make huge progress, and just the scientific discovery is like limitless when you're like really working with a strong genetic foundation. But please, like, don't forget like the silliness and the joy of what we do. Like, there's a reason we're not in agriculture and maybe growing wheat, right? Like, there's a reason we picked horticulture as opposed to, I don't know, running a combine 24/7. Like, there's a joy in what we can bring in new fruit flavors and diversity to the world. Please come see us. Like the apple and grape tour is in the fall, usually associated with the plant breeding tour if you're within that group. And then the tart cherry tour is maybe mid-July. We picked a firm date, and then all the cherries ripened before that date. And so stay tuned for maybe a more updated like tour date for the tart cherries. Have to acknowledge, wonderful colleagues at Cornell, fantastic team with the USDA, and I will leave some time for questions.
Mark, I have a question. Yeah. So you said Montmorency's been around for like 400 years.
Yeah.
And so the industry is really locked into that variety. So if you did come up with something, or we came up with something that's even better, and maybe they already have, would the industry adopt it, or are they so stuck on Montmorency that they're just not going to change?
Yeah. Talk to Amy Izzoni, who was the So Marvin's question was like, okay, Montmorency's old news, there's maybe something interesting. What's the likelihood for an industry to adopt that? And I think Amy Iazzoni at Michigan State University, a longtime breeder for rootstocks and advocate for new tart cherries, like had some success in getting Balaton and other kind of heirloom landrace cultivars into production. But yeah, just strong resistance. And I don't know, again, if there's just the risk of, it's already such a small industry and the extra effort to go to a new cultivar is not worth that to them. I think it just needs maybe a few more crop failures through frost damage, right? And because that's an easy one to overcome with some of the diversity that we have already. And just bloom late, just bloom a week later, and you've saved yourself an entire loss. So yeah, I don't know what holds back industry from that. Yeah, Chris.
Just out of curiosity, a little while ago, we had an email exchange about the apples of New York, volume 1, volume 2. How many of those apples have been saved, or has the majority of them been lost to history?
Yeah, so how many, so Chris is referencing and maybe you shouldn't broadcast this, that you have the apples of New York books, volume 1 and 2, which again, careful, there are no break-ins, but these are, yeah, so these are Hedrick, so in the early 1900s, published these Apples of New York's book. And I don't even know, I've never seen one. I've never like looked through it to see like how many are in there and how many like would cross-reference back to the collection. But it's been reported that there are, tens of thousands of apple cultivars that have been named that were at one time in the world, right? And whether those were synonyms, pseudonyms, right? Like same name, but yeah. Or like how many of those actually exist today in the collection? That's kind of hard to figure out, yeah. And we don't necessarily want all of them. So right now we had hit that acquisition phase and we filled the acreage pretty quickly, and now it's really targeted. Like we want proof that it's something really interesting genetically or culturally important or a wild accession that deserves that spot. Otherwise, yeah, we're happy to work with partners like Seed Savers or other heritage groups to conserve those, because there is quite an heirloom hobby enthusiast group for apple that's out there chasing them, and you'll read about it in the news, the lost apple groups. And yeah,
it's like especially for something like apple, where the center of origin is overseas, like you can't just like, for germplasm, you can't like shove seeds in your pocket, not that you should do that anyways, but like you obviously, is it sort of like a through university, like researchers, you exchange things? Are there similar germplasm collections overseas? Is there like a diplomacy thing?
Yeah, so there are international treaties that we're a part of that like allow for that exchange through a shared material transfer agreement. So everything's, again, above board. We're not going in and taking anything that doesn't belong to us, right? Like making sure that countries that have the germplasm maintain that ownership of it. With apple and with grape and tart cherry, it's challenging because of viruses to import. And so we do have like APHIS, the Animal Plant and Health Inspection Service, like has a quarantine site that specializes in palm fruits and stone fruits. And so it can go through there. We actually, from those 90s explorations, there's still a Malus seversii in quarantine that still has viruses that like just will not get cleaned up, right? So this is like 30 years now in quarantine. The pipeline now with like genetic tools and like screening for viruses can streamline that a little bit so you can get it quicker. But yeah, so the process for importation is a challenge. Yeah.
Well, I was going to say, when you get a chance, check with Geneva, see if Geneva has.
Let's see. Oh, so Ira, who's a new postdoc with the Grape Genetics Research Unit, asks about PCA ancestry for grape. And I'm not sure if it works for different ploidy. I mean, I assume that it would. Like, we're able to capture that and intelligibly, like, I don't know, just looking at the principal component analysis, say that like these results match what we know about these accessions, but maybe there's some flaw in doing the quantification. We'll chat, Ira. That's a good question.
I may have interrupted the question that was about to be asked.
Bill, can I ask a question? Hello?
Yes.
Yeah,
Go
this is
ahead.
Larry Smart. Great talk, Ben. Just a very quick question. I saw you have a 20K SNP chip. I'm just curious, all the genotyping that you've done on Apple, for example, was that based on a SNP array, or what are your favorite genotyping methods?
Yeah, so the SNP array is phasing out, and it was developed for commercial apples or domesticated apples, so it falls short for us. And when you start looking at hybrids or wild, we've used a lot. So my experience has been more with genotyping by sequencing, which again is every platform is flawed, but this one at least can account for different genomes, and it's updatable. So as new genome references come out, or you have a new wild accession that you don't know where it fits, that's been really helpful in kind of elucidating relationships. Collaborators now are just going for full genome sequencing, and so that'll probably be the future. But I think disentangling the pedigrees of Apple, the SNP array, and just its reliability for the domesticated has been really helpful for that data set.
I'm supposing what you distribute from the center varies, right? In some cases, it might be seeds. In other cases, it could be wood. Perhaps.
Yeah, so what do we distribute? It's primarily cyanwood because people want to propagate an accession, but pollen as well, or fruit samples for different studies. Seeds, not so much unless they're for wild, just because people want to maintain that genetic integrity of their accession. Seeds are nice because, well, in theory, we can trade them more easily because of virus issues, but I know some recent research at Cornell Agritech has kind of made us question that, whether it's truly virus-free through seed.
Anything else? Okay.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.